I'll tell you something about lifespan. We don't really like lifespan studies
Right.
particularly because everybody can extend the lifespan of a mouse. Honestly, if you take the mouse out and just pet it a couple times a day, you'll get a longer lifespan. And so that tells me that it's pretty unreliable data. What
Right.
I'm more interested in is being able to measure physiological changes that tell me not just that I'm keeping them alive longer, I want to see that I'm making them younger. And that's
Right.
what we've done. I mean, our immune system data is nobody's ever shown that. We are taking their immune systems and we have made them the equivalent of 30 years younger. And we've shown how you could do that in human beings. We could do that in human beings right now. There's nothing stopping us. It's just a matter of getting through the trials and figuring out where to get all the platelets.
Hello, Tom. You are CEO and founder of Mitrix Bio, so a company developing mitochondrial therapies to extend lifespan and health span. So it's great to have you on the channel, and thank you so much for joining us today.
Thank you. I'm glad to be here.
Thank you. Thank you, Tom. So Mitrix is developing this therapy based on the transplantation. I think it's transplantation of mitochondria. But before we get into kind of the therapy, more about mitochondria and what the solution is trying to address. So one of the things is if you're going to transplant mitochondria, so one of the things is they need to be able to get into cells, right?
Right.
And most
Right.
people think that mitochondria are just kind of resident within cells. So
That's right.
how are they going to so what is normal mitochondrial behavior like, and how are the mitochondria going to get into the cells?
Well, I mean, that is actually the key question. And so in the last 10 years, there's been a revolution in mitochondrial research. And what that means is basically, historically, if you look in your biology textbook, you think mitochondria just sit inside the cells. They're created when they're needed. They don't really go anywhere, and they're just like any other part of the cell. And it turns out that's not actually true. Mitochondria are extremely mobile within the body. In fact, they are like a fluid almost. That's the way I think of it now. They move around, they go between cells casually. They are transferred in the bloodstream in enormous quantities. And in fact, the stem cells, a lot of people do stem cell therapy. Well, it turns out that one of the primary things that stem cells do is they create the stem cells come over to other cells that are in need of help, and stem cells generate large quantities of what they call exosomes, which float over and go into the cell and contain all kinds of materials. Well, probably the largest proportion of those exosomes are mitochondrial exosomes. It's a mitochondria with a little membrane around it, and the stem cell is acting as a manufacturing site, building new mitochondria to donate to cells. Okay. The other thing that stem cells do is they create tiny little nanotubes, and they'll actually connect to the cell with a little tube, and they have videos showing the little mitochondria just marching down this tube. It's really interesting, right? And the bone marrow is creating enormous amounts of mitochondria every day. I mean, as we're sitting here, millions and millions, billions of mitochondria will be transferred through your bloodstream into the rest of your body from your bone marrow. Okay. And so this is this new world, and lots of people working on it. It's growing like at this point explosively. All the universities are jumping in. Grad students are excited. Of course, grad students always want to go after the newest thing. And so it's a very fast growing field.
So there's the mitochondria coming out of the bone marrow sound like they're coming from your stem cells, and they're like
That's right.
replacement mitochondria.
Okay, so yes, when I say they're coming out of the bone marrow, they're in platelets. Okay, your bone marrow is generating 100 billion brand new platelets every day on average. Okay, and there's about a trillion platelets in your bloodstream, and 100 million are, so every 10 days, at the end of 10 days, that 100 million new platelets are thrown away. They're recycled in the spleen, okay? Or they're used to create clots at some, if you have a cut or something, that's what uses to create a clot. Before those platelets are recycled in the spleen, the last thing they do is they squirt out their mitochondria in extracellular vesicles, and there's generally about 5 of them on average. And those mitochondria are inside extracellular vesicles that are coated with receptors. And by the way, this is our, I'll just say, this is our theory of why things are happening. We do know that it's proven that they come out as in these extracellular vesicles, which we call mitelets. Okay. We think that they have receptors on them that cause the nearby immune cells, white blood cells, other platelets to basically absorb them immediately. Okay, because the mitochondria is a very, very valuable cargo. It's mitochondria take a huge amount of energy to make, and so if a cell can get one for free, that's a big win energetically for the organism. So it's your body is always trying to recycle your mitochondria because they're expensive.
And mitochondria are made by fission, so it has to be. They're not like built from scratch. They come
Well,
from fission, or are they
they're
both?
built by the cell. The cells assemble mitochondria. They can be made by a thing called mitochondrial biogenesis, where the cell just says, I need more. And it goes and it finds whatever mitochondria it has on hand, and it makes duplicates of those. Or when the cell itself replicates, it will replicate the mitochondria at the same time that it replicates the cell, okay, and divide them.
Do we?
So
Yeah.
I was just going to say, yes, you do need a cell. The mitochondria can't replicate themselves. They don't have any machinery for building anything. All they do is they just run. They do have their own little tiny DNA inside them, but that tiny DNA is used, we think, pretty much exclusively for just managing the operations of the mitochondria. And the mitochondria, of course, is the powerhouse of the cell. It's got thousands of tiny little, what are basically jet turbines. They're little molecular spinning jet turbines all around the outside of the mitochondria. They burn glucose and oxygen, and they spin 5,000 RPM. They really are literally jet turbines on a molecular scale. So it's pretty amazing. They're amazing little things.
They are, and yes, the whole Oxford system is amazing. It's kind of, I mean, it's a diversion, but it's kind of strange. Most of the proteins from the mitochondria are now made in
That's right.
our nucleus, but for
That's
some
right.
reason we have never been able to transfer the proteins that actually do the respiration. Only the bacteria can do that. It's kind of strange.
Well, I think that my guess is, the mitochondria have been around for 1 billion years in our
Hmm.
bodies, right? We are built around them, okay, literally. Um, that's why people say, well, how is it that mitochondria can travel so easily? I say, because they were here first. They built us. They have the master key. That's really the easiest way to look at it. They cross the blood-brain barrier. They cross any barrier they want. I think that they really, to some extent, many of the people in the mitochondrial field have begun to realize that they're really in charge. They just built us so that they could go out and explore the universe. So we're just robots for mitochondria.
Cool. Um, okay, as you said, they have DNA, and so
Yes,
one of the issues with uh, as we get older is that this DNA, because their repair and I guess maintenance is not as good. So
right.
what happens to mitochondrial DNA? I mean, why is this system breaking down as we get older?
Yeah. So Of course, there's been all these theories of mitochondrial aging for 40 years, and people have had all kinds of things where they say, oh, it's ROS or it's, various other things that are causing mitochondria age. But the thing to remember is that the mitochondria itself is rebuilt every 100 days. And so the mitochondrial membrane, it's rebuilt anyway. So it's hard to imagine that that really is the cause of aging. The thing that is eternal, that is not rebuilt, is the mitochondrial DNA. The mitochondrial DNA is a tiny little ring. It's similar to a bacterial DNA. Generally, there's about 5 of them per mitochondria. So they're actually spaced along the inside of the mitochondrial membrane. And they're constantly, and they're tiny, they're 16.5 kilobits of data, kilobases, whereas our nuclear DNA is 8 billion kilobases of data. So obviously the mitochondrial DNA is this teensy-weensy little thing. And all it does is it just runs, as far as we can tell, it just produces a couple key proteins that are really need to be produced inside. It would be too inefficient. Remember that mitochondria are designed for optimal efficiency. If you're a rabbit and you're running to try to get away from a wolf, you live or you die based on the efficiency of your mitochondria. So they are optimized every way possible. And so one of our philosophies is, however mitochondria are built right now is probably at 99.99% as good as they could be built. They've been so well optimized by nature. So,
so you've
looked. So you were asking about what goes wrong.
Yes.
So those little mitochondrial DNA in there, they're replicating constantly, of course, because as mitochondria are replicated, the mitochondrial DNA are replicated. And the problem is that as they're replicated, they begin to get transcription errors. That is our theory, and quite a few other people now. The DNA itself, the mitochondrial DNA, acquires like bad sectors on a hard disk. Okay. If you remember the old hard disk where you could look and you could see all the little black dots where the bad, and the computer has to work around all that, right? Well, that's our theory, is that there's bad sectors on the mitochondrial DNA because it is such an intense environment. It's a factory. It's very hard to do quality control. It's very intense. This is happening. It's like it's hot. It's, you could imagine trying to do something careful when you're inside of a steel mill and there's fire everywhere. I mean, it's a very difficult environment. And so we start out, when we're babies, we start out with a huge complement of absolutely perfect mitochondrial DNA. And then as the decades go by, they get more and more and more degraded from these replication errors. And according to our research, by the time you're about 95 years old, they're shot. They can't support you anymore.
Right. Yeah, actually, we were talking to somebody else who said that the mitochondria actually run like 5 or so degrees hotter than the rest of the body.
Because they are literally burning. They are, as I said, jet turbines. They are steam engines. They're boilers, literally boilers. So
yeah, they are amazing things. If there's one mineral you should be worried about not getting enough of, it is magnesium. Magnesium is the body's master mineral, powering over 600 critical reactions, including hormone balance, stress management, sleep quality. Even digestion is influenced by the presence of magnesium. But there are 2 big problems here. The magnesium in the soil has been drastically reduced since the 1950s and is also removed by food processing, which explains why it's estimated that up to 75% of the population may be deficient. And most supplements contain only 1 or 2 forms of magnesium when our body needs 7. The only magnesium supplement on the market that has the full spectrum of all 7 forms is Magnesium Breakthrough by BiOptimizers. BiOptimizers are offering an exclusive deal for my audience, and it's for a limited time while stocks last. Right now, you can get a bottle of Magnesium Breakthrough for free. just go to magbreakthrough.com/modern free. Thank you for your support. So you built a clock, or you looked at the rate of DNA, of
That's
mitochondrial
right.
DNA degradation, and you built a clock from that.
Right.
Could you talk a little bit about what you saw with that?
Why don't I share a screen with a diagram, and I'll just show this to you.
Yeah,
Okay.
that'd be good.
Okay. So can you see this graph here?
Yes, can see the graph.
Great. So what this is, what you're seeing here, is an actual scan of mitochondrial DNA, and this is done with what's called long chain PCR, which is it's a very new and very, very accurate way of looking at mitochondrial DNA. This is actually 500 strands of mitochondrial DNA, which are circular, that have been opened up and laid flat from left to right. And so what the left here is 0 and the right is 16.5 kilobits, okay? And each of these little tiny thin gray lines you see here, that's one strand of mitochondrial DNA. It's on each pixel. And so what you're seeing is 500 of them. And the white areas, you don't worry about those. That's just areas where the PCR machine lost some segments. And so it just puts white. This area here, where you see these, do you see these blue bars up here?
Mm-hmm. Yeah.
Those are actually genes. And so this is the area of the DNA that is encoding specific genes that are used, specific proteins that are used. This left-hand side over here is what's called the control region. And that's, nobody quite understands exactly what it's doing, but that's where all the control circuitry is for that mitochondrial DNA, okay? And by the way, just so you know, some people may not know this, mitochondrial DNA are directly handed down from mother to child with no modifications whatsoever. So you actually share exactly identical mitochondrial DNA with your siblings and your cousins and second cousins and third cousins going back hundreds of years. It's very slow change in the mitochondrial DNA. And there's 26 major groups in different parts of the world, A through Z. And, you know, the Nordic countries have their own slight changes to their mitochondria compared to the Middle East, for example. Okay. And all that happened, you know, hundreds of thousands of years ago when the human race spread out. So what you're looking at here is all these mitochondrial data. The blue dots are bad sectors, and the black lines are bad sectors. And you can see this is actually my mom's mitochondria. She's now 95. And you can see that there's quite a bit of damage in her mitochondria. Now, here's mine. And since I'm her son, I should have identical mitochondria to her. But you can see that there's nowhere near as many bad sectors. I mean, you can just by looking at it, you can see the difference, right? So that's the difference between a 63-year-old and 95-year-old. Here's my nephew on my sister's side. So he has exactly the same mitochondria, all came down from my mother. He has even cleaner mitochondria, although it's not perfect. And we have an algorithm that we've built that's kind of a learning machine learning algorithm that gives these scores. And so my mom has 25% damage, I have 11, and he has 7. If we put that on a graph, this is what we got. Okay, and of course we had to massage this data a lot to get a. It's not really straight. It actually is quite curved. But the point is that the quality of your mitochondrial DNA is degenerating as you get older. And we think that there's a certain point where it no longer, and as that happens, that means that the mitochondria is not running properly, which means it's not generating as much energy, which means that your body is just running out of juice, which is no surprise. Anybody who's gotten in their 70s and 80s can tell you that just the energy level just starts to plummet. You just don't have the energy you had, so things don't work as well. We think that I also want to mention that decline can happen much faster if you don't take good care of yourselves. And that's why people put all this effort into fussing about their diet and getting enough sleep and so forth. The reason that that works is because it helps your mitochondria hang on longer.
Right. And the lack of, I guess the lack of energy is not only, it's at a cellular level, so it also impacts chronic diseases like Alzheimer's.
Absolutely. Absolutely. Our theory is that there's a certain threshold level below which you start, all these diseases start to pop up. And above that, your cells can kind of always just push through, right? Like, heart disease. Well, your cells mostly when you're in your 30s and 40s, they've got a lot of energy and they can keep going and they keep replicating and they can dispose of waste and they can do their jobs really well. But once they go below a certain point, it doesn't work as well. And then you start to get scar tissue and you start to get plaque buildup and all these things that begin to turn into heart disease. Same thing with Alzheimer's. Okay.
Right.
It doesn't,
Yes.
it doesn't mean that the mitochondria is necessarily the only cause.
Mhm.
We consider mitochondrial energy decline to be like this background, almost like a river underneath, and everything is affected by it in either large ways or small ways. Okay.
Yes. So Mitrix is looking to develop a therapy
To do
to
this.
to address this. So yeah. So what is yeah? How does this therapy work?
Okay. So the idea is if by, let's say by 75, 80 years old, you've lost enormous amounts of functional mitochondria. You've actually lost just a lot of numbers of your mitochondria, and you've lost your inventory of mitochondrial DNA has dropped significantly, 40, 50%. You just don't have as much as when you were young. And so what we're aiming to do is to actually restore, to replenish your mitochondria in your body. And so the only way to do that we can see, of course, there's no real source of mitochondria. You can't go around squeezing them out of teenagers because the teenagers won't like that, right? So the solution is you have to grow them. And that's why we're building a bioreactor system to grow mitochondria on massive scales. And then we've developed techniques to basically replenish your stock of mitochondria, which, as you can imagine, we're talking about somewhere between 2 and 10% increase in the mitochondria in your body. So we're talking trillions upon trillions, maybe quadrillions of mitochondria that are going to have to be transplanted into you. It's going to be quite a big effort.
Are these mitochondria taken from you, or are they generic?
Well, let me talk about that process real quick. So basically, many people now have gotten stem cell treatments. In a stem cell treatment, what do? Usually they take stem cells out of your body. Maybe they take them out of your blood, or maybe they get them out from liposuction or something. And they take the stem cells. They may or may not expand them in a bioreactor, and then they inject them back into you. Okay.
Mhm.
Very common. Sometimes they do it for joints to get your hip or your knee to regenerate. Sometimes they use them for facials. There's a lot of people doing stem cell facials now. Mitochondria really are just extracts of stem cells. So what we're talking about doing is instead, we'll take out the stem cells, we grow them. Instead of growing them 10 times, we're going to grow them 10,000 times. We're
Mhm.
talking about huge amounts of your tissue that we're expanding in a bioreactor. And then we extract the mitochondria from those stem cells. So instead of re-injecting the stem cells back into you, we're extracting the mitochondria. We put them in a coating to protect them, and then we inject them into you. And the reason to go through that process is that the mitochondria are very, very, very small. So when you extract the mitochondria from a stem cell, you're getting this tiny, tiny drop compared to an enormous amount of stem cells. And so you can do a huge amplification of the process. And the mitochondria, we believe, are the most powerful component of the stem cell. And so that's the part that you want to optimize for. Okay. So it's
Yes.
not really that different. It's not really that different from a stem cell treatment in a lot of ways.
But mitochondria would not have um, rejection issues because people take exosomes from other people, and the exosomes contain the mitochondria, and there's no rejection.
Well, actually, once you start getting into the quantities that we're talking about, and by the way, I'll mention, if you go do a stem cell treatment and you get mitochondria when you get a stem cell treatment. According to our calculations, 150, 200 million stem cells is typically what you get in a stem cell treatment. That's about 2 days worth of youth. Okay. It's too small. I've always said stem cells are a great idea. It's just not enough. It's too small. You need more. We're talking about a 10,000x amplification of that process. We want to get you 20,000 days of youth. That's 20 years. Okay. So to do that, of course, that's why we're doing this thing with extracting the mitochondria. But when you do that, there are different haplogroups of mitochondria, and there is some research to indicate that if you get foreign mitochondria from a completely different person, if you get them in small quantities, it's not going to affect you much. But if you get it in the kind of quantities we're talking about, we don't know yet. There could be the potential for cellular dysfunction. And so that's why our focus right now is on autologous mitochondria, using your own mitochondria.
That makes sense. So one other thing so we talked to other people about mitochondria, and they say that they are not homogeneous. It's like the mitochondria in your heart might be different from the mitochondria in your brain.
That's right. That's true.
Is that not true for stem cells, or how do you handle that?
Well, it's an interesting point. That's right. The difference is that the mitochondrial membranes are structured a little differently because, for instance, in the brain, in the neurons, they have to be really long and skinny, and in the heart, they need. So they're different. They're different flavors of mitochondrial membrane construction. But remember, we don't care about the membrane particularly. Our focus is on getting the mitochondrial DNA in there. Okay. So the mitochondrial DNA is exactly the same. It doesn't matter whether it's in the heart or the brain or wherever. Now, somebody probably will say, no, that's not true. Our belief is that the mitochondrial DNA, of course, everybody will argue this, and there's a lot of people with a lot of theories, and we're just one of them. So just take of course, I'm giving you my perspective. But our perspective is that the mitochondrial DNA are consistent across all species of mitochondria. And so we just say, hey, we're just going to pump you up full of brand new mitochondria, and the body will work out the rest of it. We actually think that in some cases, you might have mitochondria floating around in the bloodstream that transfer across the blood-brain barrier, and they are used in the brain. Okay. We have seen that data. I would assume that the astrocytes, which are the cells that collect materials from the blood-brain barrier, are probably modifying the mitochondria that they get from the blood in order to make them compatible with the neurons. Okay. And so I think that there is a modification and translation facilities that are built into our body.
Right. I mean, it would make sense because we only got one copy of mtDNA from our mother in the first place. Well, we got multiple copies, but
you get every egg cell has 500,000 mitochondrial DNA in it. It's quite enormous. It's almost as much DNA in volume as the nuclear DNA, but it's just 500,000 copies of exactly the same thing. Right. And by the way, that brings us back to the question. There's a guy named Nick Lane, by the way, who does a lot of research on mitochondria and has published some great books. And they talk a lot about mitochondria in the reproductive system. How do we make mitochondria young again? Because if it wasn't for that, we wouldn't exist as a species. I mean, in order to create youth, you have to have a way of making the mitochondria young again for the egg cell, right? And the answer is that it's a very energetic. The female reproductive system spends a lot of time and energy sorting through the mitochondria and finding the good ones and packaging those into the eggs, and then expanding them massively so that new baby is getting the biggest possible number of healthy mitochondria to give it the best possible start.
Yes. How often would people need to have this therapy? I mean, looking at your graph, it looks like every 20 years. Have you thought about it?
Nobody knows.
Nobody knows.
How could we possibly know?
Yeah, no, that would
Because
make sense.
nobody has ever done this. This is not in nature ever anywhere. So, we've done it in a lot of animals, and we've done it. Some people have tried it in human beings on very small scales. There are people who have saved lives with mitochondrial transplantation. It is not that new. It's been used to save children who have mitochondrial diseases, but this is only just in the last few years. So we don't have any answers about what would happen in adults.
Right. So currently, what would it look like for someone to have the therapy when you get to the clinic?
Well, let me tell you, we are planning for phase 1 trials in about a year. Okay.
Mm-hmm.
I will say a year from when we get our Series A raise, which we're working on right now. The phase 1 trials will be a group of probably 20 volunteers. We actually have most of them now. We've got a few. We're still looking for more volunteers, by the way. Most of them are in their 70s and 80s. I have a couple 90-year-olds. We did get William Shatner, the actor, to get on our oversight committee. He's 93. He's obviously interested. So it's turning into a great project. It's going to be very exciting. And the approach that we're going to take is we're going to start very, very slowly, because again, we've done this in animals. It's very hard to know whether it's going to be more dangerous in human beings, because human beings have extremely fussy immune systems compared to lab animals. Okay. So I can do this. We've done this in mice for years and gotten amazing results. We've seen mice turn younger, literally become younger, but those were mice, and mice and human beings are completely different. So you want me to describe that process that we're going to go through?
Yeah. So let me see, because you started talking about animal experiments, and I'd love to talk about those as well. But yeah. So when you envisage that this is available in a clinic, what would it look like for the person who is kind of receiving the transfusion?
Well,
transplantation?
Again, I can't give you much in the way of details, because we need to try this in some human beings first.
Yeah.
And as I said, we have this group of our what we call our explorers, and we're all going to go off and we're going to explore this new world, right? Like going down to the Antarctic and being the first to reach the Antarctic. So, but eventually our stated goal is that eventually that we have gigantic factories pumping out mitochondria by the ton all over the world. Every country has their own mitochondrial, supplies, and that they're given almost like vitamins to people over 50 years old. Okay. I mean, I think that from a standpoint of the healthcare system, if you could keep people's hearts and brains and immune systems younger for another 20 years, I mean, you could reduce healthcare costs dramatically, right?
Yeah.
So from a standpoint of a person getting that treatment, it really might be as simple as going in and getting a blood transfusion. It might look a lot like that. You go in, the doctor says, "Oh yeah, you're mitochondrial type H21A. Here's a bag of that. I got it from the factory. Here you go." Boom. And you get it, and they keep you in observation for a couple hours, and then you go home. Okay. That's probably, a while off. Probably in the short term, what we're going to be doing is we will do this almost like what's called CAR T therapy. If you know anything about CAR T therapy, we will, for each individual person, we'll go in, we'll extract their stem cells, we'll send them to a factory, we'll grow them, we'll extract the mitochondria, we'll put them in a coating, we'll send it back, and then that will be reinfused back into the person, either an intravenous injection, or we might even do it in there. A lot of the times now they do these injections in the tummy fat. Okay, they just put little needles in there, little tiny needles, and they give you a dollop of whatever the material is. So, is that going to be every month? Is it going to be once a year? I just don't know yet.
Yeah. Cool. So before we move on to the human, and I definitely want to talk about the human trial because I think that's really
Yeah, yeah.
exciting. But what have you seen in animals in vivo with, and have you done like full transplantation for them? Like you've tried
Oh
to
yeah.
replace
Oh yeah.
significant.
Absolutely. Oh yeah. We have papers published, peer-reviewed papers, and many other people have too. I mean, as I said, there's dozens of teams all over the world doing this in vivo and in vitro. Okay. So we have a group that is doing in vitro work, and then we do everything in parallel. We then go do it in mice. And so we're doing in vitro and we're doing it in vivo at the same time. And so for example, the published paper we did last year, we took mice that were considered to be, elderly, 12-month-old mice, 12 to 18-month-old, which is middle-aged to elderly. 12 to 18-month-old mice, their immune systems are significantly depressed at that point. And so we had a group that was very expert at testing animals by giving them a case of sepsis, which is a bacterial infection. And you can tell, how long they're going to last. And so we had a group of mice, and we cut it in half, and half of them we gave all of them sepsis, and then half of them got no treatment, and the other half got these mitochondrial injections. Okay, what we call mitelets, which are mitochondria that come from the platelets, and those were donated from young mice. We got platelet transfusions from 1-month-old mice. We got enormous numbers of these mitochondria from their platelets, and we put those into the elderly mice who were sick. And the net result of that is that they survived. And it was a quite exceptional result. It was quite exciting. So it was the equivalent of going to a hospital and finding someone who's in the ICU with COVID or pneumonia, who's 80 years old, and let's say you go in, you give them a transfusion of young mitochondria, and what you're doing in effect is you're making their immune system 30 or 40 years younger at that point. It's a dramatic effect, and it's really quite simple.
Yes.
It turned out to be much simpler than we thought it was going to be.
Have you tried it in kind of healthy old mice? And do you
Yes.
see
Yes.
rejuvenation? Have you tried
Yes.
lifespan?
Yeah, absolutely. So we did it in mice and looked at their brain to see if we could see improved brain tissue, and we saw that. These are mice, they're even by 12 months, they have a dramatic drop in the mitochondria in the hippocampus, which is the center of memory. And mice, and of course, they can't do the mazes, and it's very well studied. And we took those same people who studied mice, and we did these mitochondrial injections, and we saw a significant increase in the hippocampal mitochondrial expression, what they call gene expression. Then we did a bunch of injections into mice. Actually, they're all still alive. We're continuing this. They're up to about 23 months old now. We took a bunch of mice and we just give them every week. We give them a big dollop of 1-month-old mitochondria, and they don't seem to age, quite honestly. You see increases. We saw a significant increase in the ability for them to run on the wheels, where we saw that their weight changed quite a bit. Usually mice, as they get older, they get really fat, and these mice continue to have what you call lean muscle mass. So they appear and they look younger. So,
Are you going to release like a lifespan paper based on
yes,
that?
Yeah, absolutely. But I'll tell you something about lifespan. We don't really like lifespan studies
Right.
particularly because everybody can extend the lifespan of a mouse. Honestly, if you take the mouse out and just pet it a couple times a day, you'll get a longer lifespan. And so that tells me that it's pretty unreliable data. What
Right.
I'm more interested in is being able to measure physiological changes that tell me not just that I'm keeping them alive longer, I want to see that I'm making them younger. And that's
Right.
what we've done. I mean, our immune system data is nobody's ever shown that. We are taking their immune systems and we have made them the equivalent of 30 years younger. And we've shown how you could do that in human beings. We could do that in human beings right now. There's nothing stopping us. It's just a matter of getting through the trials and figuring out where to get all the platelets.
Right.
So
Yes. Yes. The immune system seems to be the base of so much of aging, and
yeah,
so being able to
I mean,
rejuvenate it.
just think if we could make all the 80-year-olds, if we could, when needed. And by the way, that's temporal. It only lasts a few days. So, the vision is that you use it as a therapy. You get them out of the hospital. You get them to go home. And then eventually it wears off and they go back to being who they were before. But for a couple of weeks, they have a young immune system and they can beat off these infections. Okay. So you can imagine the impact on the healthcare system if you could just do that.
Yes. Okay. So speaking of humans,
Yes.
you have started this trial, and I think you call it like the 130-year project because
Yeah.
you're aiming to beat the 122. That's the
We
current
are
limit.
aiming to beat the 122-year lifespan. Exactly. And now obviously we won't know that for a while.
Hmm.
The oldest person we have is 90 years old who's a volunteer. So it may take us 40 years to prove it. But if we can show, I don't know, are you familiar with the Healthspan X Prize?
Hmm. A little bit, yeah. If you could provide a little bit of background on it,
Yeah, the
you're aiming for that.
Healthspan X Prize was sponsored by the Saudi Arabian government, and it's the X Prize Foundation is running it, and the goal is it's a $101 million prize, and the goal is to show that you can take 10 people and make them 20 years younger. Okay, and of course everybody says, well, what exactly does that mean?
Hmm.
You know, it's not like you can just put somebody on a treadmill and get some exercise and say, oh, they're 20 years younger. That doesn't. You have to figure out a way to prove that you've made them physically and genetically younger, right? Which is challenging. But that's our goal. Our goal is to show that we can take our 20 volunteers and make them 20, 10, 20, whatever years younger so that they have increased muscle mass, their brain improves, their cognition improves, their heart regenerates, their immune system gets better, that they are literally younger. Okay.
Okay. And when are you going to start this trial?
We've already started the early phases. As I said, we've gotten the volunteers. We decided to get a group of volunteers early because we wanted their help in kind of helping us determine how to do this. And when you're working with 80-year-olds, you're getting people with a lot of experience, right? So I have a couple really, really good doctors who are in their 80s who are still practicing doctors who are experts in gerontology, and I'm getting the benefit of their experience for this project. So that's really exciting. Um, the goal is to start it in a year to a year and a half from now. That would be 2025. But we are in the process of raising the money to do that. You need significant funding to do something like this. This is not a biohacker project. You need a lot of big bioreactors and they're very expensive.
Right. And it is the funding, it's not approval from any regulatory agencies that's the limiting factor.
No, we're probably going to do this in either Japan or the Bahamas because both of them have specific mechanisms in their regulatory rules that allow for these sorts of trials. Japan, for example, if you get through the phase 1 trials, which are very rigorous, I mean, you have to do a good job. And we're going to do. We have some of the best scientists on the West Coast and East. We have some of the best scientists from all over the world who are going to be supporting this project. So we have to do this very high quality. We want this data to be usable by anybody. This is a we want this data to apply to the whole human race. Okay, so we're doing it very well. In Japan, if you go through the phase 1 trials and prove safety, then they'll let you actually sell it to the general public as part of the phase 2 trials. They have a different system there where they use the data from early customers as part of the trials. And that's also a way that makes it easier for companies to get past the funding hurdles because they can sell things early.
Interesting. Yes.
Yeah. That has been, by the way, that system has been proposed for the United States and I think is being seriously considered.
Right. Interesting. I think it's kind of going back a little bit, but I saw on one of I saw you talk once about, so we have our DNA in our nucleus, right? And we
Right.
have partial reprogramming that we can do to recover that to some degree.
Right. Right.
But if you do that in an old cell and you've got old mitochondria, then that's still not going to solve the problem. So have you thought about the combination?
Oh yeah. No, but that's our if you look at our kind of standard deck, which you can, we have YouTube. We have our presentations are on YouTube. One of the things we always talk about is what we call the laptop analogy. You can tell I'm an old computer programmer because I was talking about laptops and hard disks. The laptop refurbishment analogy. And, whenever you take an old laptop and, you send it off to some factory, they refurbish it and they resell it. That's a thing that people do. Whenever you get an old laptop, the first thing they do is they put in a new battery and a new power supply. They don't even mess around. They don't test it. They just put in a new battery. And the reason is that you need the power to do anything else that you want to do. If you don't have good power, you can't rewrite the operating system that you need to rewrite, okay? And so we think that mitochondrial transplantation and epigenetic reprogramming absolutely should go together because you need the mitochondrial DNA fixed, and then you need the nuclear epigenetic DNA fixed, okay? And they're working together. Remember, the nucleus and the nuclear DNA and the mitochondrial DNA, they're literally co-computing. And that's been a lot of research about that. The mitochondria actually serves as a sub-processor for the nucleus. And there's hundreds of them, and they're scattered all over the cell. So you can imagine, the nucleus is using the mitochondria almost like subsidiaries. They're out there, and we think there might be a lot of sensory input that the mitochondria are collecting and sending back to the nuclear DNA. So if you don't fix both of them, you're never going to get the really high quality restoration. So that's my argument. I think if I personally, if it was up to me, I'd do both for sure. For me personally.
Yes. Yeah, I'm just wondering because so far in vivo reprogramming has not been that successful. They've had some success, but not that much.
It's hard. It's very hard.
Sure.
I absolutely hope and pray that they succeed. I think it will take years, and there's a lot of money going into it. That's why we think that mitochondrial. I always say mitochondrial restoration is maybe 50% of the problem, but it's really the first and it's kind of the low-hanging fruit. Like we could do it right now.
Right.
Hopefully, that's everything we've seen so far makes it appear that it is fairly simple and straightforward to do. It's hard to grow the mitochondria, but transferring them in is easy. It's not easy, but it's doable. And so I would say that's the thing I could do in the next few years. And maybe I only get people another 10 years of life. Well, that's okay, because then that gives them time to wait for the people to finish the epigenetic reprogramming work and get that working properly, right? So
Yes, I saw that. So one thought is it would be nice if the people who are doing the epigenetic reprogramming in mice could also do mitochondrial transplantation at the same time. But the thing about extending people's lifespan, yeah, that kind of plays into the longevity escape velocity
Right.
idea.
Exactly. And my argument is mitochondria are the first thing to do, right? Because you boost their energy levels. Also, if you've got someone who's 80 years old and their cells are already chronically energy depleted, all this other stuff is just going to be hard to do because everything takes energy, right? Even epigenetic programming, it takes energy. The cell has to grab the vector and it has to go over there and it has to translate it and blah blah blah. And it's just going to be so much easier if we can re-energize people first.
Yes,
That's philosophy.
that sounds good. So we kind of talked about the future bit. So I mean, the idea of having off-the-shelf mitochondria that you could just go and get
Right.
injected. I just
Right.
wondered, what do you see is there anything else that you would like to talk about in terms of the possible future for mitochondrial transplantation or mitochondrial therapy in general?
Absolutely. Let me just say, I mean, we've talked about longevity quite a bit, but remember that mitochondrial transplantation has got this whole other world of just being a disease cure that is actually where most people are using it. So longevity is kind of the exotic use for this, but the very prosaic and very kind of off the shelf normal thing you could do with mitochondrial transplantation is just, cure diseases. So if you, as I said, if you have people who are elderly, who have an infection, let's say they have sepsis, which is very dangerous. Well, all right, give them new mitochondria. Boom, get them out of the hospital. That's a really easy win right there. Uh, the other one is eyes, for example. We've shown that you could really cure a number of eye diseases, retinal diseases. And that's quite simple because it's so small. So you don't need very much. And so that's work that we were doing at Stanford, for example, where we've been testing uh, diseases like AMD, which is a blindness, a disease that causes blindness. You could do. There's people who have published papers where they show tremendous improvements after heart attacks. So emergency medicine, obviously, like every emergency room should have a couple big buckets of mitochondria in their freezer, right? And you come in there, you've had a heart attack, boom, they give you mitochondria right away, and it just restores, reduces the amount of dead muscle tissue that is left behind after that. Anything where there's any kind of strokes, tremendous opportunity. Obviously, you could use this in the military for battlefield injuries, for radiation poisoning. Okay, radiation poisoning is primarily a mitochondrial process. Uh, Chemotherapy kills mitochondria by tremendous, is what chemotherapy does. It literally is designed to kill the mitochondria. Well, what that does is it leaves people with this post chemo. You've probably heard about that post chemo weakness. You just can't do anything for years, and it's very destructive. So there's a lot of, and there's also a lot of childhood diseases, by the way. There's children who are born with mutated mitochondria, and obviously that's the first thing you want to do is try to repair childhood diseases. So we've talked about. I just want to say we've really pushed this idea of sending mitochondria to the moon. Okay, if everybody wants now, they want to go to do these lunar missions and they want to go to Mars. You should be taking mitochondria for the astronaut.
Yeah, for the higher radiation that they will have.
Yeah. And
Yeah.
and it's also for wound healing. What if you fall down? People still fall down on the moon, and just they
Yeah.
could fall down and get a big cut. And you don't have any doctors nearby and you're there for 4 weeks. And maybe you give them some mitochondrial stuff that you smear on there that helps them heal faster. So
So do you have a separate kind of track that's looking at these more limited applications rather than just the 130-year project for? rejuvenation.
Yeah, we're doing both simultaneously. So the 130-year project, we created a subsidiary called the Biotech Explorers. And we stated the 130-year project is, we've said actually it's for astronauts. That's our first group. We're doing our volunteers, and then because we're not sure how fast you could expand this. So who should get it first, right? Well, I always argue, the astronauts risk their lives, so they should get the longer lifespan, right? And then after that, we'll have to figure out, how you can get it out there. So that's that. And then our other part of our company is working on these other indications.
More target. So very briefly, I mean, what kind of timescale for human trials on these more targeted applications?
I don't know exactly.
Yeah,
That is, okay. It's all based on funding. So
okay.
The biggest issue, I mean, we're a tiny company. We don't have any kind of big offices or labs or anything. We have a whole bunch. We got the best researchers in the world. They're all working for stipends and doing this research. But you can't expand this without getting the funding. And unfortunately, this is so new. It just takes time.
Yeah.
The biotech funding field is very conservative because it's very high risk. So it takes time to get their attention.
Right. But it seems very promising in that it's seems very safe, quite simple. And although mitochondria are not well understood, at least that we're getting better at that, but it's not like jumping. I mean, I can see that partial reprogramming is a lot harder to convince the FDA that this is a good idea. But mitochondria seems simpler.
Yeah, but it's interesting because there's been a lot of mitochondrial startups. Interestingly enough, Bill Gates, for example, is a big mitochondrial fan, and he's funded mitochondrial startups that have, but they're all using chemicals to try to enhance the performance of the mitochondria. This idea of just replacing the mitochondria, it's like I always tell people, it's like when your car battery goes bad in your minivan, right? And it's always the battery that goes bad because the engines last forever. Do you try to put new fluid in the car battery and try to restore it, or do you just take it out and put in a new one? Okay. And what most good car mechanics will tell you is, if the battery goes bad, you just put a new one in, right? And I think that's really kind of this new philosophy that we're coming up with is, since it turns out that it's relatively easy to install new batteries into cells, why don't we just manufacture brand new ones instead of messing around trying to fix up the old ones? Okay.
Yes. Yeah. Especially you can't fix the DNA, the mtDNA with chemicals. DNA.
I mean, it is. I mean, this is going to be very expensive. So, I mean, what I always tell people is, I don't think this is going to be for people under 50. I don't think it'll be worth it, at least in the beginning, because it's going to be very expensive. I think the best thing you do is eat well, don't smoke, live healthy, and protect those mitochondria because they are the only ones you have, okay, until we figure out how to replace them in large quantities.
Yes. Okay. And on that note, where can people follow what Mitrix is doing and the 130-year project?
Well, go to our website, mitrix.bio, mitrix.bio. Also, my LinkedIn account, I post a lot of, it's more or less like a blog. And so I post a ton of stuff on the Mitrix LinkedIn account and on my own personal LinkedIn account. So just find us on LinkedIn.
Excellent. I will do that. And we will link to those in the description. So, Tom, thank you so much for joining us today. That was really interesting.
Yeah, my pleasure. And thanks for spreading the word. It's for us, it's all about educating people about the magic of mitochondria.
Excellent.